The script that's written in our genes directs us from behind the scenes. The words within it shape life's destiny. Hidden in your DNA is your genetic dossier. It tells your future and your history. How traits get passed from parents to a child is something that has kept us so beguiled. Cracking the code, genetic mysteries to unfold. Cracking the code, genetic secrets will be told. Cracking the code, genetic mysteries to unfold. Cracking the code, genetic secrets will be told. Cracking the code.
After an exciting race that ended in 2001, 2 different versions of the human genome sequence were published. Although they both had small gaps to fill, between them they included almost all of the 3 billion letters written within our Book of Life.
Reading out this instruction book is a wonderful milestone, but as you then start turning the pages, you realize, I don't have a clue what this means.
A lot of people viewed getting the genome sequence as the end of a process. I argued all along it was the beginning of a process, that it was a race to the starting line.
The first task in understanding our Book of Life is locating and cataloging all the genes buried within it. Not only is a gene a small island in a much larger sea of DNA, it is broken up itself into even smaller bits called exons, which are separated from each other by larger bits of apparently inactive DNA called introns. Introns were discovered only after the invention of DNA sequencing in the late 1970s. They came as a complete surprise to scientists at the time and overturned their concept of a gene.
So that a gene is spread out over a relatively large part of the chromosome. It was a very dramatic change in our thinking.
So it is a challenge to find the genes and parts of genes in, essentially a sea of 98% other bases that in some way are noise.
The easiest way to locate genes within these billions of letters is to look for the particular 3-letter codons that signal to the enzyme that transcribes the gene into messenger RNA where to begin and where to stop.
Well, the start of a gene in the human genome always starts with an ATG sequence. And then you have a long stretch until you get to the end of the gene, which has a very definitive stop sequence. There's different combinations, but one can actually scan the sequence to identify these features. In between, there are specific nucleotides that tell the enzymes that copy the DNA sequence when to splice the DNA. So just by using computer scanning programs, one can identify very accurately where the start, stop, and what the constituents are in between of a particular gene.
And that's useful, but it probably will only identify maybe 3/4 of all the genes.
So other methods are used to find the genes buried within our Book of Life. One of them came out of a surprising discovery about how this genetic information is transformed into biological action. As formulated by Francis Crick in the 1950s, the central dogma of genetics held that genetic information flows from DNA to RNA to protein, and never in the opposite direction. Then, in the early 1970s, it was discovered that certain viruses contain an enzyme called RNA transcriptase, which can reverse this flow so that RNA is transcribed into DNA in violation of the central dogma. Using this enzyme, geneticists were now able to transcribe any messenger RNA sequence into its complementary DNA sequence, which could then be read out like any other piece of DNA. The base pairing rules dictate that this new DNA sequence matches up exactly with a DNA sequence from within the gene that produced the messenger RNA in the first place. A computer scan of the entire genome can easily locate this unique bit of DNA. Once found, it tags this location as the site of yet another gene. This method of finding a gene by working backwards from its RNA product was pioneered by Craig Venter, who later went on to head the privately sponsored Human Genome Project. He called it the Expressed Sequence Tag, or EST method, and it is now an essential tool for gene hunters.
It's actually still the #1 gene discovery tool in the world. It also created the whole genome industry.
But the EST method can only find a gene if it is actively expressing its messenger RNA.
The muscle gene, for example, you have lots of muscle, so it makes many copies. So you get lots and lots of copies of that. Whereas a gene which is used once, maybe, in the womb to make a particular bit of the brain is extremely hard to find because its copy will only be there transiently and in very small amount.
So it varies tremendously from tissue to tissue. You can't just take all the ESTs and count up and say that's how many human genes there are.
So geneticists rely on yet another method to identify human genes. It compares our genome to those of other species with whom we have shared parts of our evolutionary pathway.
Take mouse and human for an example. You may not think they're very close, but they're both mammals. That means they both have the same body plan. And they diverged some tens of millions of years ago, and since that time, they've gone on their separate way, except for those key bits of the genomes that are essential for making a mammal. And those bits will stay the same. And so now, if we bring back the two genomes and line them up, we can see the bits that are the same and the bits that are different. And so this is like a spotlight which illuminates the key bits of the genome.
Obviously, a yeast cell, very much like a human, nor does a worm, nor does a fruit fly. But if we look at their genes, we find we're amazingly closely related.
A lot of the genes that are needed to make a cell grow and divide were worked out 1.5 billion years ago and are common to every organism on this planet.
The genes that are involved in replicating DNA, the DNA polymerases, for example, they're almost identical in sequence between bacteria and human. And the reason for this is the sequence is so essential to the life and the composition of that organism that it had to be maintained through evolution. And it worked for bacteria. There's no reason for nature to change it. It should work for human, and that's really why it's been maintained.
All the various methods of finding the genes in our genome rely on computers. Many biological experiments are now done using a different kind of mouse.
Well, there are now biologists who never touch a living thing. They touch keyboards and they process information. They, of course, work with other biologists who are working on living organisms and experiments, but you can make remarkable progress by studying the data.
If you ask me, the field of biology that is most in need of new creativity and energy and good people is the computational side. And we need well-trained people who understand biology really well, but also understand computer science really well.
Geneticists at their computers are now able to analyze and compare entire genomes, a fast-moving new field of discovery called genomics.
It's just the most extraordinary trove of information. It's as if we've been living all our lives in a vast library and we didn't realize there were books around us and we couldn't read. And suddenly someone turned on the lights and we can see books upon books on the shelves, and we've just worked out how to read, and we're going to be up every night for centuries reading these books in the library.
We have now located and cataloged almost all of our genes, but that is just the first step in understanding our book of life. The next is to figure out what those genes are all doing. The function of the genes is to produce proteins, and so proteins are the focus of the next great effort at understanding our book of life. Genes code for proteins using the uniform 4-letter language of the nucleotides, strung out in a one-dimensional sequence. This information is then translated into the non-uniform 20-letter language of the amino acids, which leads to a 3-dimensional sequence.
What's interesting about this 20-letter language is it has many different sizes and charges and shapes. And this enormous diversity has the property that the order of these 20 different letters in the protein language dictate how that string folds in 3 dimensions to make a molecular machine. And it is that 3-dimensional structure that allows it to execute its particular kind of function.
The complex structure of these molecular machines can be studied on a computer but not their function. To study protein function, geneticists must do biological experiments using living organisms. To study a protein experimentally, geneticists often start with the gene that produces it, and then manipulate that gene in some way or another. They can splice it into a single-cell organism like a bacteria. As it multiplies, so too does the foreign gene. The foreign protein it produces can later be harvested from this culture and then analyzed. The function can be better studied in more complex creatures like roundworms. First, its gene is inserted into a fertilized egg cell, which otherwise lacks it, and then the effect of its protein is observed, as this starter cell multiplies and develops into an adult organism, each cell of which incorporates this new gene. The function of a native gene can also be studied by altering it through deliberate mutation. This will produce an altered protein, which in turn may produce an observable change in function. This can give valuable clues about the function of the unaltered gene and protein, especially if the mutation is precision-made.
If you change the gene at one place, that will change an amino acid in the protein, and then you see, does that change of amino acid make the protein work differently?
In the late 1970s, Canadian scientist Michael Smith invented an ingenious method to produce these very precise mutations. It makes use of a surprising discovery he made about DNA probes, the short bits of custom-made DNA which geneticists use to locate a target sequence such as a gene. The single-stranded probe bonds to its single-stranded target because their opposing base pairs match up with each other according to the base pairing rules. Smith and his team discovered that if this bonding occurs at a lower temperature, the probe will still stick even if one base within it is a mismatch. Using this mutated probe as a primer and the target strand as a template, he was able to produce a complete strand containing this one-letter difference. During replication, this mutant strand produced its own double helix, which now fully incorporated the single-letter change. Smith called this new technique site-directed mutagenesis. This precisely altered gene could then be inserted back into its host cell, usually a simple model like a yeast, to express its slightly altered protein.
And these are often genes which are important from the human health point of view. For instance, there are genes in humans which cause cancer, and there are often analogs of those genes in yeast. Not that yeast gets cancer, but you can study the molecular properties of those genes very well in yeast and learn things about them, and then go and apply that knowledge to human biology.
Site-directed mutagenesis revolutionized the study of gene function, and in 1993, it won Michael Smith a Nobel Prize in chemistry.
And I feel very, very lucky because I wasn't wise enough to plan this. My getting into DNA was essentially an accident, but I very much appreciate that it was a wonderful accident.
Even after we discover a function for each of our genes, there will still be much to learn about our book of life.
These are just the first steps of biology. Almost all the complexity of biology comes after the genetic code. That's the simple part.
This complexity is first introduced after the gene is transcribed into messenger RNA. An editing process then takes place, during which most of the introns are spliced out. The remaining bits come together in various different combinations, each of which represents a different RNA message. Further complexity comes after these messages are translated into protein. Each different protein can then be chemically modified in a number of different ways, which again multiplies the number of different proteins that can be derived from the same gene.
Some genes hypothetically can code for maybe 1,000 different proteins. So with 25,000 different gene elements, each that can code for multiple different proteins that get changed further in the cells in different environments, it becomes quite complex to work out all the different combinations.
And you may find the primary function, and you may be 100 years before you find all the others that they're all connected with. So it's no less and no more than the total understanding of the total organism or all the organisms in the universe before you're all done.
Up till now, the only way to gain this knowledge has been through experiment, an often tedious and frustrating business. But in the future, there may be a much quicker and easier way to figure out the function of a gene.
If there's kind of a holy grail to genomics, it would be being able to take the sequence of a gene and predict the three-dimensional structure of the protein that it encodes, and then say that because it has that three-dimensional structure, it's a gene that metabolizes cholesterol.
In the future, we hope that's true. That'll be one of the biggest breakthroughs in biology, but we're trying to learn some of that language. We don't know how to predict a structure of a protein very well just from looking at its linear sequence.
When the human genome was first sequenced, the biggest surprise was how few genes we have. Not a whole lot more than the tiny roundworm that geneticists love to study. The main difference is that our genes combine together to form networks, groups of genes that interact with each other to produce an endless variety of entirely new, more complex effects.
And so we have this finite set of genes that in various combinations create all the biology we know.
Because we're not just talking about genes taking up individual jobs, like this gene's in charge of your left toe. We're talking about amazing amounts of combinatorial interaction between these genes.
So another great challenge will be to work out which groups of genes work together to produce the kind of complex protein chemistry seen in these time-lapse films of a cell. Genes that are active at the same time in the same cell are often part of the same gene network. So an important first step in uncovering these networks is learning which genes are expressed together within the same cell. Messenger RNA provides the best window into gene activity. Since it's a short-lived molecule, its presence reveals that the underlying gene is currently active. And because its sequence is complementary to that gene, it can also reveal its identity by tagging to it and then sending out a signal of some kind. So by studying which messenger RNA molecules are present in the same cell at the same time, geneticists can begin to work out which genes work together in networks. To do so, they now use this tiny device called a gene chip, which has revolutionized the study of gene activity and gene networks. It is a piece of glass onto which a precision robot deposits thousands of different bits of single-stranded DNA, each taken from a different gene and specific to it. Therefore, each matches up with one RNA message and no other. The robot is programmed to lay down each bit of DNA onto its own precise, predetermined spot on the glass. Once assembled, the chip is exposed to the contents of the cell being studied, including its RNA messengers, which have all been tagged with a light-emitting marker. Those that match up with DNA strands on the chip will hybridize and stick to them, while the rest are spun away. The gene chip then goes into a scanner, which looks for the light-emitting markers. Each square on the screen represents a different gene, and the precise location of each square identifies that gene. Those squares that are lit up by markers represent genes that are actively expressing their messenger RNA in the cell being tested. These are genes that are probably interacting with each other in the same gene network. So far, we have regarded our book of life as a single manuscript that we all share in common. But because we have two sets of chromosomes, one from each parent, we actually carry two slightly different versions of it. And each of these versions is slightly different from everyone else's, except in the case of identical twins. It is these small differences in DNA that set us apart as individuals.
Our visual system is very good at recognizing subtle differences in facial features, for example. Chimpanzees probably think we all look alike, but these differences all go back to small differences in the genetic code.
So a full understanding of our DNA will also require knowing how it varies between two matching chromosomes, and then between any two individuals on Earth, and then learning what effect all this genetic variability has on our biology.
It's genetic variability that makes some of us tall and some short. It's genetic variability that makes some fat and some skinny, and that makes some predisposed to diseases like cancer.
What inspired the Human Genome Project in the first place was our desire to find those DNA variations that underlie human disease. Some genetic diseases are caused by a mutation in a single gene. Cystic fibrosis, for example, but these are the exception.
Most diseases and complex conditions, even aging, are not the product of a single broken gene, but of the interplay of many, many genes. These are called complex traits. They also become susceptible to analysis if you have all the genes at your disposal, because you can start to see which ones are varying or interacting in particular individuals or families to produce a particular disease state.
In fact, almost all the chronic ailments that plague us are due to the combined effect of a number of gene variations, which in turn interact with lifestyle and environmental factors. But these groups of genes are far more difficult to track down than the single mutations that cause simple genetic diseases, like cystic fibrosis. That's because the usual family groupings used to link an unknown gene to its closest DNA marker fall short when searching for gene networks.
Since if we have 100 genes contributing each to the disease equally, so 1%, then it might be difficult by linkage analysis with a reasonable number of individuals, with few hundreds, let's say, individual, to pinpoint where we have a linkage. This is below the resolution.
So to establish these linkages, geneticists look for an isolated population whose children have no choice but to marry within the group, and who therefore have a high incidence of certain mutations, including those that cause a complex disease. One such group is the Cochin Jews of Israel. For thousands of years, they lived in the city of Cochin in southern India, surrounded by other religions. To maintain their Jewish identity, they married only among themselves, which added a genetic component to their isolation. Then in 1954, almost the entire community emigrated to Israel, where they continued to intermarry.
And here in Israel, they developed asthma in very high frequencies. Between 40 and 50% of the individuals suffer from either asthma or allergy or both. This is really high in the general population. Also in Israel, the frequency is between 5 to 15%, depend on the population. So above 40 is really, really high.
There is now a worldwide effort to find the multiple genes that underlie asthma. But Sheva Karam is leading a team at Hebrew University in Jerusalem that is searching for this network of genes among the Cochin Jews.
We think that this is a perfect community to try to identify the asthma genes.
They have collected a study group of over 1,000 Cochin Jews with proven asthma and worked out their huge and interconnected family trees. They will then test each member of these families with hundreds of DNA markers representing every region in the genome. The presence or absence of these DNA markers in this unique population will then lead them to those regions that contain the asthma genes. They will then know from the human genome sequence which particular genes to focus on in search of these mutations.
Once we identify the networks which are affected in asthma, we can think about how to fight them. Without this knowledge, we might not have the best therapies.
But isolated populations like the Cochin Jews are hard to come by. So gene hunters are now turning to large groups of unrelated people who share a particular complex disease. Since the DNA linkages are so weak in such groups, their plan is to do away with markers altogether and to just sequence and compare all of their DNA.
You can imagine having 1 million genomes of people all with one type of cancer, another 1 million with a certain type of heart disease, another with schizophrenia.
Right now, there's no way to do that. It would be expensive and too difficult to do it. But I don't think anyone has ever gone broke betting on the capacity of molecular biology to solve technological problems. Molecular biology is always technology-driven.
Sidney Brenner, one of the founders of molecular genetics, has an even bolder plan.
We want to take people, a lot of people aged 5 to 50, and follow them through life and see what they eat and what they do and look at their genes.
Instead of beginning with a complex trait and then tracking down the genes that cause it, his plan would take the reverse approach. It would start with a full knowledge of all the genes and the environmental exposures of thousands of people, and then observe which traits develop from which genes over the course of time.
See what diseases they get, what diseases they don't get. And if we find something, then test it on 2 million people.
Whichever approach is used, we will eventually uncover all the genes involved in all of our most common diseases.
All of these are going to have their contributions unraveled because we have these tools now to do that.
Once these complex disease genes are identified, it will be easy to test for their presence in any individual. These new genetic tests will then lead to a new kind of medicine.
That is, we'll be able to look at this variation and say to you, these are the diseases that you're likely to get in the future and that you should be worried about.
Now, those won't be yes or no predictions. They'll be relative risks, probabilistic statements. You'll be able to learn, oh, your risk of colon cancer is 4 times the average. That could be very useful. We know how to keep people from dying of colon cancer. It's to make an early diagnosis. But we don't do that in everybody because it's too expensive, and many people aren't too thrilled at the kind of procedure they might have to go through, even though it's not that bad. A lot of people consider it sort of unattractive from their perspective. Well, if you knew for you that you had a 20-30% chance of dying of colon cancer unless you go through this process, you'd probably be pretty motivated to do it.
Medicine will profoundly change. Its whole focus will be on keeping people well rather than attempting to make you well after you get sick.
And that will be very powerful. A lot of the things that get to us are things that could have been avoided if you knew what to do.
Other ones won't be so cheerful because they'll say, well, you may well have a predisposition to this or that disease, and at the moment we don't have a treatment for it.
But new treatments will also flow out of the discovery of new disease genes.
Every one of those is going to point you towards a pathway that must have gone awry in this disease. And that in turn will tell you, oh, if that's the pathway and this is what's wrong, how could we tweak it back in the right direction? And those tweakings will basically be the next generation of pharmaceuticals. So by, say, 2020, we'll not only have preventive medicine to focus on keeping you healthy, if you fall through the safety net and get sick anyway, we're going to have things to offer you that are profoundly more effective than what we have now for most diseases.
I won't be around at that time, but, you know, people have asked me, what would you like to do? I said, gee, you know, I'd like to be able to come back just for one day, you know, just on a day trip from wherever I am, just to see what's happened.
Our book of life has now been cracked open. The great challenge ahead is to understand all that it has to teach us about ourselves.
But along the way, I think light bulbs will be popping on and off all over the place as people begin to realize what you can do with the whole book of life in front of you instead of having a page here and a page there, which is all we had before.
Cracking the code. Genetic futures will be foretold. Cracking the code. Genetic power about to explode. Cracking the code. Genetic mysteries to unfold. Cracking the code. Genetic grids will be told. Cracking the code.